This figure is showing the development of an egg from fertilization to the 2-cell, 4-cell, 8-cell, and ultimately to what's referred to as the blastocyst stage. The blastocyst will go on to ultimately become the embryo, and from the embryo the complete organism will derive. So what preceded the 2-cell stage was the 1-cell stage, or the zygote, and the zygote was produced by fertilization, in other words, union of the sperm and the egg. That's followed by mitosis. So one diploid cell produces two diploid cells that are identical. These divide to produce 4 cells. These divide to produce 8 cells. And then we have a series of events that ultimately lead to the formation of the blastocyst. And again, the embryo will form from this particular portion of the blastocyst. Now, cloning is the production of genetically identical individuals. So natural cloning occurs when, in the early stages of development, the cells will actually designate themselves to become different complete organisms. Remember, each of these cells contains the complete genetic information to make an identical cell, or to ultimately make an identical organism. So what you can do is isolate fertilized eggs that have just started the process of dividing. What you can then do is remove, or split, I should say, split these two cells, and there's the opportunity for each of these cells to develop into a complete organism. You can also do that at the 4-cell stage. However, as the number of cells increases, the technique becomes increasingly more difficult to accomplish successfully. So this was probably the earliest form of cloning, which is called embryo splitting, where you take the embryo, you separate the cells, and ultimately each of those cells develops into the complete organism.